It's
Yeah.
amazing. Cool. So let's get started. Hi, everyone. Thanks, Olaf, for inviting me. Thanks, everyone, for joining this Methods Day. It's also my first Methods Day, so I'm very excited to be here. I'm going to give you this brief talk about information theory tools for cognitive neuroscience. And before we start, I would like to acknowledge the people that are part of this work, mostly Fernando Rosas from the Center for Psychedelic Research at Imperial College, Dan Ball from the Department of Psychology in Cambridge, and Andrea Lupi from Clinical Neurosciences and UVA, as well as a bunch of other people from University of Sussex and Imperial and Cambridge. So being in consciousness and cognition lab, of course, one of my main motivations in doing all of this work is actually defining and measuring consciousness, which is a pretty big deal and it's kind of tricky. But to do that, actually, one of my main sort of motivations at a sort of almost metaphorical level, actually, I take inspiration from thermodynamics. In particular, I was very heavily influenced by this book called Inventing Temperature. Um, and I actually, at some point I came to the realization that actually consciousness science now is essentially where thermodynamics was about 200 to 300 years ago, where they were facing the problem of inventing temperature and thermometers at the same time, which is a weird sort of scientific chicken and egg problem. And I think in consciousness neuroscience now we're in very much the same situation where we're trying to define consciousness and measure at the same time. And we need to do this iterative process of refining our measures and our theories iteratively. So with that in mind, my goal for a whole research program in general, and what I'm gonna, the brief um, snapshot I'm gonna present today is basically to do a theoretically and empirically informed revision of our tools to measure consciousness and cognition. And more specifically, this involves answering questions like, what are our tools actually measuring, and does that match what we want them to measure, and can we improve them in some theoretically informed ways? To do all of this, my framework of choice in particular is information theory, which is not really committed to seeing the brain as a information channel sending bits around, but more generally, it is a very convenient way to study and summarize statistical structure of multivariate time series. And in particular, one sort of very high level starting point I'd like to start from is that the information or the entropy in a signal can be split. So what's going on in the present can be split into these two terms, one that I'm going to call inheritance, that is information that comes from the past, and some innovation, which is the new information that is coming in at every sort of sample in the time series. And I'm going to sort of take this division, I'm going to split the talk in two, and I'm going to first talk about this innovation, or what's mathematically called as the entropy rate. And I'm going to say how this is useful for neuroscience, how it's been applied, and how can we compute it. And then I'm going to talk about the inheritance term, which is a mutual information. I'm going to do the similar thing. So again, this talk, I'm going to divide this in two parts. First, I'm going to talk about entropy and the particular estimator of entropy called impulsive complexity. And in part two, I'm going to talk about information decomposition synergy, and I'm going to give some brief links to integrated information theory. That's a very prevalent theory of consciousness, a very well-known theory at least. All right, so let's jump in very quickly to the topic of entropy and complexity. So the entropy rate is this very fundamental object in information theory that basically measures, how unpredictable is a particular time series. And one very not canonical, but say simple estimator of this entropy rate that's also very effective is this thing called the impulsive complexity, which is very, very widely used in computational biology in general and in consciousness neuroscience in particular. And the way this impulsive complexity goes, or the way it works, again, very, very simply, is if you have a sequence or a string of discrete symbols, you can like split it into parts and find what different patterns make up this sequence of symbols. And if you have a complicated sequence, that you may have to split up in many different patterns, whereas if you have a very simple sequence, you may be able to split it in just a very small number of patterns. And this means that one of these sequences has this very high impulsive complexity, and the other one has a very low impulsive complexity, which again is related to how easy it is to predict. You can see that the lower sequence is very, very easy to predict because it's always A's. And you can do a very similar thing, actually, but with, for example, EEG signals or any other sort of neurophysiological signal. And one of the canonical examples is that if we sort of take a REM and non-REM sleep neurophysiological signals from human subjects, that in general complexity goes down when people are unconscious, say during non-REM sleep, and it goes up when you're awake or when you're in your REM phase. And in more generally, this implicit complexity represents complexity as a diversity. So you're not looking for a particular pattern of activation or a pattern of activity. You're looking for you're estimating how many different patterns are in your signal. And this approach has actually turned out to be very, very successful in consciousness. So this is, for example, one very seminal paper by Casali and a bunch of others in Science, Translational Medicine, some years ago, that basically they applied this measure to EEG, and this turned out to be a remarkably good estimator of, or a remarkably good index of conscious level. So it is very high if participants are awake, and it's very low if participants are either asleep or under various forms of anesthesia. And not only this impulsive thing goes down when you're unconscious, but it can also increase under certain conditions. So, for example, we had this recent preprint where we showed that actually not only this impulsive complexity goes up when you're on a psychedelic drug like LSD, but it also goes up when you're sort of receiving some external stimulation, like watching a video or listening to music. So this all goes to show that LIMPUS is actually a very effective tool to study consciousness cognition in general. But it has two important limitations that I'd like to address, and that deal with that are addressed by the methods that we have developed recently in the Department of Psychology. One is that there's an unclear relation between l'impulsive and the power spectrum of those signals. So you might ask, to what extent, let's say we see a difference between the l'impulsive complexity of an awake and a sleeping subject, to what extent is this difference driven by the known spectral differences in power between the two conditions? And the other question is that, or the other issue, is that l'impulsive has very limited spectral and temporal resolution. So you need like a relatively long chunk of data to estimate this thing properly. And I'm going to very briefly mention a couple of methods that we have devised to overcome these limitations. One of them that we have in the archive at the moment is this decomposition of the l'impulsive difference between two conditions. So if you have two data sets and you can take the difference in this l'impulsive quantity, or actually any nonlinear function for that matter, via doing some funky phase randomizations and phase swappings between the signals and their power spectra, we can actually decompose this total difference of l'impulsive into three different terms. One is a term that is due to, the difference is due to the phase in the Fourier sense of the word. Then the differences are due to the power spectrum, and the differences are due to the interaction between the phase and spectrum in both conditions. So for example, we applied this to the MEG data in the CanCan dataset, in particular comparing rest versus a stop-no-go task. And what we can basically do is decompose these differences that we observe into these different types of effects. So if you look at the left panel, that is the total difference in l'impulsive complexity between rest and task. So we see a sort of apparent frontal decrease in complexity, whereas complexity increases basically everywhere else throughout the brain. But actually, if we tear this apart, we can see that the spectral effect and the phase and the interaction effects have very, very different topologies. In particular, some of them focus on the sort of frontal side of the head. And this basically gives us another sort of degree of interpretation of the data. So we can say, well, l'impulsive changes in, according to some topological distribution, but it does so for different reasons in different parts of the brain. So we have this extra resolution, or this extra nuance to our analysis. And then another limitation of l'impulsive is that so far all examples rely on spontaneous activity. So l'impulsive is seen as a state measure, have some long recording that are split into chunks and compute and then average, because you need these very long recordings to estimate this thing properly. And as a target application, we wondered if we could use this limpulsive or something like limpulsive to study complexity in fast processes like the ones we typically see in cognitive neuroscience. So as a target application, we started looking at mismatch negativity in the primate brain. So it's a very standard paradigm that some of you may be familiar with. So there's some public data in the NeuroTico database of monkey ECOG. And the monkeys are just listening to these tone trains that have some standard and some deviant stimuli. And the standard result that you see at the bottom right is that there's this ERP signature that is much stronger in the deviant than the standard stimulus. And basically, we developed this new method based on a family of models called state space models that allow us to basically compute this instantaneous limpulsive complexity type measure. So we can evaluate this measure of complexity at every time point throughout the sort of response to a single stimulus. And we get this very interesting result, actually, that in this auditory oval task, the entropy changes precede the ERP by 20 milliseconds. So by using this temporal decomposition of a complexity measure, we have basically been able to find a signature of this mismatch negativity that precedes the standard signal by about 20 millisecond difference. And we have replicated this across a bunch of animals, and we're currently undergoing more studies to see how well this generalizes. So far, I've shown you a few methods and a few sort of applications of how to study entropy and complexity in MEG, or in this case, ECOG signals. Now I'm going to switch gears a bit and talk about information decomposition and mutual information for the last few minutes of my talk. Mutual information, which again is something that some of you may be familiar with, is a general measure of independence between two variables. That's a sort of generalized version of the linear correlation coefficient. So if the mutual information between, say, x and y is 0, that means they're independent. Otherwise, it means they're somehow statistically dependent on each other. And one very natural application of this is basically a pairwise functional connectivity matrix. So if you have a brain, you measure some form of multivariate time series from it. In this case, the example is MEG. Then you can basically look at pairs of regions and compute their correlations or their mutual information and build this matrix of pairwise dependencies. And these are being used for a very, very long time in neuroscience, as I'm very sure you know. However, there's an issue, which is that the brain is actually a lot more than just two interactive parts, and things are a lot more complicated than they seem at first. And as a toy example, let me show you this tiny system of 3 rings. These are called the Borromean rings. And they're very interesting because if you, in your mind, try to visualize how this thing looks if you remove, say, the red ring from this system, you will see that actually the blue and the green are decoupled. And this applies to any of them. So this is, a group of 3 sort of variables or 3 rings that are coupled only when they're seen as a whole, but not if you see them in pairs. So if you were to compute the functional connectivity of this thing, that's all zeros. There's some form of dependency, there's some form of structure that you only see at the high order, but you don't see if you look at the pairs of elements in the system. A natural question emerges, and we can ask, can we decompose information that is shared between more than 2 variables in a multivariate dynamical system like the brain? The answer, you'll be shocked to hear, is yes, through this method or this framework called partial information decomposition by Williams and Beer in 2010. And in this PID framework, what they do is basically split the mutual information that a bunch of variables have about each other into 3 terms mostly. Redundancy, unique information, and synergy. The redundancy is the information that is held by all variables jointly. The unique information is information that's held by one variable in particular. And the interesting bit, in our eyes, is this bit that we call synergy. And the synergy is the information that is in the whole, but it's nowhere in any of the subsets of the system. So it's in the whole, but not in the parts. I know this is the EEG session, but I would like to mention here an example in synergy and redundancy in fMRI, led by Andrea Lupi, where we find actually that synergy is associated with high-level cognitive function. So it's a lot higher in frontoparietal networks, and they correlate very well with some neuroscience meta-analytic gradients that you can find in this recent preprint that we put up. And then finally, for the last minute or so, I'd like to make a brief sort of introduction, very, very brief mention to integrated information theory. That's a theory of consciousness that says that, or that claims that consciousness occurs, or a system is conscious whenever its parts integrate information, the whole of the system integrates information to a larger extent than the parts. And to do that, there's a bunch of integrated information measures called phi that typically what they do is compare a system with a disconnected version of itself. So in these little diagrams, we have two-time series with time going from left to right. And we somehow cut the system, and you see how much those two statistical models differ. And with PID, what we can do is we can zoom in into the constituents of phi. And we can see that phi is actually not a single thing, but it's an aggregate of different things. And in particular, that these other normal definitions of phi are sort of a weird mix of synergy and transfer and redundancy and many different things. So what we can do using this framework is to formulate a revised phi, or a phi-er, that includes only the terms we like, such as synergy and transfer. And with those, we find also some very interesting results, namely that this revised version of phi-er, but not previous versions, decrease consistently in the default mode network and the propofol anesthesia and disorders of consciousness. So I'd like to wrap up on that one. And as a sort of very quick summary points, what we have developed in here is a bunch of new tools for impulsive complexity analysis that are more efficient, versatile, and nuanced. We can use this partial information decompositions that reveals what kind of information is being processed by a multivariate system. And we found some links with synergy with high-level cognition and consciousness. And we've also used this to get new insights on IIT and causal convergence. There's, of course, many questions open, such as extension algorithms, applications, and more. My hope is that if you found any of these methods interesting or possibly useful for you, then I'm very happy for you to approach us and then, or go and read the papers and see if there's anything that we can do to apply these methods to your data, or collaborate in some other way. I'm going to flash some references there real quick, and that'll be the end of my talk. Thank you very much.